As I mentioned at the outset, part of our goal in our symposia are to highlight the people that you all interact with when you come in, our nurses, our PAs, our faculty, our other associates. But part of the goal really is to highlight the unsung heroes, the people behind all of what we do, who enable all that we do going forward. So it's really a pleasure to introduce Mary Horak, who was my first data manager. How many years ago? I can't even admit how many years ago that was, but who has really been a core of the database system at the Hillman, in the melanoma program, in other programs, annotating the tissues, which I mentioned to you before, are the issue. The tissue is the issue because without it, we don't understand what is happening in the tumor, as you heard from Dr. Najar. And all of that data either makes the tissue useful or without it makes it useless. So Mary, thank you for coming to talk. I think you'll all learn a little bit about the heavy lifting that goes on behind the scene. And Mary, thank you for doing it.
Thank you. Hopefully I live up to that wonderful introduction, so thank you. So I am here to talk to you about tissue banking and data, and I am on the clinical side, so the lab is definitely not my forte, but I hope I can bring this together for you. So to start, what is tissue? So tissue is a group or layer of cells that work together to perform a specific function. So we might think of our skin as a tissue. However, we also have other things in our body that are tissue that aren't just tumor samples, for example. So we have blood, urine, stool, all of those are considered tissues in our research. And these samples are also often referred to as biospecimens or correlatives. So why is this important? So you all know tissue is important to make a diagnosis. So what stage it is, those different features, but we also use tissue to find ways to prevent, diagnose, and treat cancer. So tissue research often takes a long time. We need to gather tissue from a lot of patients to compare it. And then we also store this in a tissue bank for future use. So why are tissue banks important? So tissue banks are facilities that organize all of this tissue. So they work to collect it, they store it, they distribute it to other researchers for this science and for this study. And we can use tissue for basic science and then also translational science, where we're going from bench to bedside for treatments with our patients. So by studying tissue in the lab, we're learning about biomarkers, we're learning about molecular signals that our prior presenters have talked about, and we're learning about how cancer cells work and how they might respond to different treatments. So basic science often begins with this tissue that we store in tissue banks, which again leads to the development of these new treatments. So how are we using these tissues? So tissue is used to determine how well patients respond to treatments. And looking at that, we're also able to determine how well that treatment works. So we can also work to develop a theory and then use this tissue to support that theory to determine if it's correct. And research with molecular mutations help us find treatments that we can target to your specific mutations. So BRAF, NRAS, KIT, these are all mutations commonly found in melanoma. So we can use the tissue to drive the research to find those targets and those treatments. So how is tissue analyzed? So one of the things we look at is the tumor microenvironment. This is something Dr. Najar talked about, and the tumor microenvironment is the cells within the tumor, also the cells surrounding the tumor, feeding the tumor. So all of that around your tumor is part of that microenvironment. So it's really important to look at that because we can look at individual markers within the tumor itself, but the whole microenvironment as well. And there's multiple techniques we can use to analyze these tumors. And then we also, each technique gives us a different view. So we need to look at them collectively so we can see that big picture of what's going on in that tumor. So looking at on the left side of the slide, the bottom row is flow cytometry. So this is a way where we can look what cells are present. So it shows us, are these tumor cells? It shows us what cells are present. So we don't necessarily know if they're tumor cells or if they're normal body cells. But the confocal microscopies, the top two panels, that shows us what's going on in the tumor. So is it alive? Is it active? And we can look at these different microscopy and combine them together to get that overall picture. So with the orange arrow, the bright blue means this tumor is active and alive. So looking at these different things collectively, we know which cells are active, which cells are metabolizing and causing cancer to grow or perhaps spread. The one thing about flow cytometry is we can also use it on blood. So that is something where we can use that to learn more about metastasis and how the treatments might work in the blood. So we can also analyze tissue to look at biomarkers. So these biomarkers, again, are something that Dr. Najjar talked about. We can look at groups of patients to see, does this subset of patients have a response? And this can allow us to develop targeted therapy. So this is an example of this kind of science that led to the PD-1 treatments by looking at how these biomarkers interact. So we can also use tissue to determine treatment response. So the slide on the far left is normal tissue, and then the slide in the middle is a complete pathological response to a treatment, and then the far right is a pathological non-response. So what is about this patient in the middle that their tumor responded? And this work is actually from Dr. Najjar as well. So kind of looking at this in the last 3 slides, first we started looking at the tumor itself, then kind of patients that have certain expressions. And now we have a particular patient, how did they respond? So with that, we can develop these clinical trials. So the goals of clinical trials are really to develop treatments for our patients. They are written by researchers and biostatisticians who ensure that we have measurable outcomes that we can analyze. So we want to make sure it's something that we can compare to really get, is there something statistically significant happening? So these objectives and endpoints are the scientific questions we're aiming to answer. And these objectives drive the data that we need to collect. And safety and efficacy, so is the treatment safe and how well is it working, is commonly primary objectives for our clinical treatment trials. So again, safety is assessing adverse events. So those are the side effects. And then efficacy is the disease response. So is the patient's cancer getting better, staying the same, or getting worse? So what data do we need to do this? So clinical characteristics are really important because we have to look at these in conjunction with the tissue analysis to determine the effectiveness of the treatment or the nature of the cancer. So this is something Dr. Kirkwood referred to. We need that big picture. Otherwise, you just have tissue and you can't correlate it to anything. So common data collection information includes demographics like gender, race, age, the date of diagnosis, the cancer and tumor characteristics, mutations, prior treatments, medical history, baseline problems, medications taken, because we're looking at this to, again, look at patients to kind of tease out which ones, why are we seeing the responses we're seeing. So for safety, the adverse events, when Melissa was talking about, all those common side effects we see, this is the research that allows the investigators across the country to say, these are the side effects we expect to see in our patients. So for any of you who've been on our trials, your nurses probably have gone through your side effects and asking all of those probing questions of how tired are you? Are you able to, get dressed? Are you able to make your bed? We use a tool where we grade these based on a very standard system to make sure that we are assessing all the side effects the same way, and we can assess them across different patients on a particular trial. And then our physicians are the ones who are determining what the cause of that side effect is. So is it related to the treatment or is it related to some other cause? So if a patient falls, did they fall because they're clumsy and they tripped, or did they fall because their treatment made them dizzy and they fell for that reason? So that's how we're assessing these side effects for safety. For efficacy, we're looking at how the disease is responding. So is it staying the same, getting better? So if you remember in Dr. Najjar's slide, she had an image of scans with patients' disease. It looked like maybe a liver of different sizes. So this is how we correlate this. So we measure the tumors on those images and we compare them from baseline. So on the left, we have a baseline scan. So our measurement is 3.37. And on the right, we have a follow-up scan. So that's 2.3. So that disease has gotten smaller. So we have a standard system we use again to determine what that response is. And in this example, would be a partial response. And this, again, is how we're determining how the patients are doing. So we compare this across all patients on that study to determine the overall effectiveness of that treatment. So what do we do with this data? So we have to put it in a database, and there's many different ones that we use. Medidata Rave is one, Inform is one, REDCap. So these are common databases that researchers across the country use. And in this example, we're just kind of seeing the snapshot of the different fields that we're collecting. So we have baseline information, so past treatment, past medical history, if the adverse events we're collecting, so all those side effects we're seeing, concomitant medications. So are these patients taking other medications? Are you taking metformin or Tylenol? We like to look at that to see, again, if there's any correlations. Response, follow-up, all of that information we're capturing. One thing I wanted to note, we always capture this information by a subject ID. So any patients in our trials, we aren't putting your name in these databases. It's a unique number that we're tracking you by. So what else can we learn? How can we put this together? So while the main objective of many of our treatment trials are safety and efficacy, secondary objectives are often involving tissue research. So these objectives are used to determine how the treatment is changing the body's immune system or the tumor itself. And then I should note, we do have studies that these tissue kind of based objectives are the primary objectives, but in many of our studies, they're also the secondary. So the goal of these objectives, again, is to forward the science so we can develop better treatments or understand even why certain treatments aren't working. So what patients do not see treatment responses with. So again, how does this come together? So the data from the tissue analysis is correlated with the clinical data that we were looking at. And the biostatisticians are the one that help us kind of do the analysis and see, are we seeing trends? Is it more than just random chance that we're seeing these results? So the analysis can be conducted grouping these patients differently based on their characteristics. So, do men do better than women for a particular treatment? Does age matter? Also looking at those tissue analytics, so molecular mutations, gene expression, biomarkers. Again, we can group these patients in many different ways to really tease out which patients are having the most benefit from these treatments. And then once these results are reviewed in the context of all of these objectives, the tissue, safety, efficacy, whatever the endpoints might be, we determine how well that treatment worked. So that leads to new treatment approval. So when statistical analysis shows that a drug is safe and effective or meets whatever endpoints, we can submit that to the FDA for approval. And the FDA thoroughly reviews this data before they make the approval for safety and accuracy. So our work directly contributes to these approvals. So the trials that you as patients have participated in have led to this. So we have a trial with Dr. Kirkwood, who in January 2022 was approved for uveal melanoma, and then one with Dr. Luke for our stage 2B and stage 3C patients with Pembro. So again, our research directly makes a difference in the landscape of cancer treatment. So why give us tissue? So hopefully I've explained all the important reasons why you should give tissue. So it really contributes to the advancements of science. This helps find a cure. So you are helping us learn more about cancer. It allows you to take an active role in your healthcare. So you learn more about melanoma and the different treatments. And then trial participation is always voluntary. So it's always at will. And then just lastly, a huge thank you to all of you, because without you, we don't have our treatments. So thank you. And I have some references. Any questions?